Hello everyone, this is Dr. Santosh. Today we will be talking about catabolism of protein and gluconeogenesis because of that catabolism. So this catabolism of protein occurs during prolonged fasting condition. Actually, the metabolism of protein is tilted towards anabolism during fed condition, but during fasting condition, especially prolonged fasting condition, what happens is protein the metabolism of protein is tilted more towards catabolism. So let's talk how does this protein catabolism occur. Actually, we are not going to talk in very detail about this protein catabolism. We will talk in a broad, we will tell a broad view of protein catabolism. We will discuss a broad view of protein catabolism. So as we know, the protein is made up of amino acid, the chain of amino acids. So linked by peptide bond. So what will happen? Protein through proteolysis it will generate amino acid. So with proteolysis it will generate amino acid. An amino acid is a compound which is made up of amino group and acid group. Amino group and acid group. And after amino acid releases this amino group, so it will release amino group as ammonia. And after releasing this amino group, this amino acid will be left with the acid group and keto acid, this keto acid group. And these all keto acids are organic acids. They are acidic in nature. And so what will happen with this ammonia? Ammonia is very toxic, and that is why ammonia will be converted into urea through urea cycle. And if there is problem in any enzyme of urea cycle, it will cause urea cycle defect. So urea cycle defect is related to protein catabolism. Now what will happen? This organic acid will be through its catabolism, amino acid will go steps. Through those various steps they will produce glucose and ketone body. Glucose and ketone body. We have 20 amino acid in our protein. Among those 20 amino acid, 18 amino acid will produce glucose. 18 amino acid will produce glucose. Those amino acids are called glucogenic amino acids. Those amino acids are glucogenic amino acids. And 80 except leucine and lysine, all amino acids are glucogenic. We have 20 amino acid, 18 amino acids are glucogenic except leucine and lysine. Both starts with L. So you can remember formula L square. So except L square, all amino acids are glucogenic. This is very important point. Now this some glucogenic amino acid, among this 18 amino acid, they will also produce ketone bodies, especially aromatic amino acid, phenylalanine, tyrosine and tryptophan. They will produce both glucose and ketone bodies. And we have two more amino acid. One is branch chain isoleucine, other is hydroxy group containing essential amino acids. So isoleucine and threonine, they will also produce both ketone body as well as glucose. So we can easily remember this formula as PITTT: phenylalanine, isoleucine, tyrosine, threonine and tryptophan. So these amino acid produces both ketone bodies and glucose. So what is the purpose of this production of glucose? So what happens during fasting condition? We don't have glucose as fuel. So our body is switched on fat for utilization of fuel. And this fat, our body, muscles, especially muscles and other peripheral tissue, can utilize fat as fuel. But brain for few days cannot utilize fat. It will utilize glucose only. Especially RBC. RBC has no choice except utilizing glucose. Therefore, a certain glucose level above 50 mg per deciliter has to be maintained in our blood. For that matter, this protein will be catabolized to produce glucose. So ketone body is also fuel source. Ketone body can be utilized by peripheral tissue as well as in after few days 2-3 days, brain also starts utilizing ketone body. So this ketone body and fatty acid become the main source of body. And for few tissues, for example RBC, glucose will be always be the source. Therefore, the maintenance of minimal level of glucose is always required. And that is the relevance of this formation synthesis of glucose from protein. And this whole process is called gluconeogenesis. This whole process is called gluconeogenesis. Gluconeogenesis. So through gluconeogenesis from protein, glucose is being synthesized. Few amino acids, they will produce acetyl coenzyme A, and that acetyl coenzyme A will convert into ketone body. And fatty acid that is produced during, that is the major source of fuel during this condition, will also produce ketone body. We will focus more on glucose today. So if there is problem in this conversion of organic acid into glucose, the problem arises is called organic acidemia. So high metabolic acidosis will occur. The example of organic acidemia is, or aciduria, is propionic aciduria, methylmalonic aciduria, and glutaric aciduria. I have already done one video on propionic aciduria and methylmalonic aciduria, which are very important among organic aciduria. Now let's talk how this organic acid is, how these organic acids converted into glucose. Let's talk about this. So this is the picture showing how this organic acid, these amino acids, when it converts into organic acids, and it will produce glucose. See, these are all amino acid, glucogenic amino acids. So these glucogenic amino acid, they will produce these organic acids, intermediate organic acids of citric acid cycle. Final product is this, and this final product will go and form glucose this way. So purpose of, I want to mention one point here, the purpose of Kreb cycle during especially prolonged fasting condition is gluconeogenesis. During fat condition, the purpose of this cycle, Kreb cycle, is production of energy. But during fasting condition, purpose is to produce glucose, and that is from the glucose is produced from these amino acids. So what happens? We will have to remember which amino acid produces this intermediate. There are 5 intermediates of Kreb cycle. If you can see 5 intermediates of Kreb cycle, and which amino acid produces what, we will have to remember. So there are, we can see this, we have to memorize glycine, alanine, serine, tryptophan, threonine, and cysteine. They produces pyruvate, and pyruvate will convert into oxaloacetate, and oxaloacetate will convert into phosphoenol pyruvate, and phosphoenol pyruvate will produce glucose. And this arginine, glutamine, glutamate, proline, and histidine, they will produce alpha ketoglutarate, and alpha ketoglutarate will produce succinate, and succinate will produce fumarate, and fumarate will produce oxaloacetate. And this is how phosphoenol pyruvate and glucose, this is how this amino acid will produce through this pathway glucose. And valine, isoleucine, methionine, threonine will join this Kreb cycle through succinate, and succinate will ultimately be converted through fumarate, and fumarate oxaloacetate, and phosphoenol pyruvate and glucose. Now these two amino acids, tyrosine and phenylalanine, they will produce fumarate, and fumarate will convert into oxaloacetate, and oxaloacetate will produce phosphoenol pyruvate, and phosphoenol pyruvate will convert into glucose. Now see this aspartate and asparagine. These two amino acid will convert into oxaloacetate, and this oxaloacetate will convert into phosphoenol pyruvate, and phosphoenol pyruvate will produce glucose. So this is how amino acid produces glucose. Glucogenic amino acid will produce glucose. They will ultimately, the product of this amino acid will be the intermediate of this Kreb cycle, and Kreb cycle during fasting condition is dedicated for synthesis of glucose. Actually, Krebs cycle is not cycle during this fasting condition. It becomes broken cycle. So what will happen? This ketoglutarate will form glucose. Oxaloacetate will not form alpha keto. It will directly go and form glucose. So this is Krebs cycle is actually a broken cycle during fasting, prolonged fasting condition and dedicated for synthesis of glucose. I have marked this amino acid, purely glucogenic amino acid, as green color, and I have marked this amino acid, the both glucogenic and ketogenic amino acid, as black color. So you can see tryptophan, threonine, tyrosine, phenylalanine and isoleucine. They are both glucogenic and ketogenic. We can see this threonine can produce succinate also and pyruvate also. So threonine can produce succinate also and pyruvate also. So PITTT. So phenylalanine, isoleucine, tyrosine, threonine and tryptophan. These 5 amino acid will produce both ketone body plus glucose. Now we will memorize this all amino acid through mnemonics. So this here is gastric. So we'll remember this amino gastric will join pyruvate. These amino acids, these are, we will give mnemonics for arginine, glutamate, glutamine and proline and histidine that will join. So we will memorize the name of all amino acids here. Valine, isoleucine, threonine and methionine, they will join succinate, and phenylalanine, tyrosine, they will join fumarate, aspartate, asparagine will join oxaloacetate. We will memorize all the name of this amino acid through mnemonics. Let me talk about this mnemonics. So this mnemonics, you can see this gastric pylorus. So pylorus is part of your stomach. So we can see we can use this mnemonics gastric pylorus. So those amino acids, G for glycine, A for alanine, the simplest type of amino acid, will form pyruvate. So gastric, remember gastric pylorus. So glycine, alanine, serine, threonine and tryptophan, they will join Krebs cycle as pyruvate, and pyruvate will be converted into oxaloacetate, and oxaloacetate will into phosphoenol pyruvate, and phosphoenol pyruvate ultimately through multiple steps it will convert into glucose. Now second mnemonics is R, glue and glumine, provided for his conglomerate. Conglomerate is our artwork where multiple different color material are pasted together. So glue is required for pasting. You can use this mnemonics like that. So conglomerate here is alpha ketoglutarate. So this R for arginine and glue for glutamine, arginine and glutamine, glutamate, and glumine is glutamine. So arginine, glutamate, glutamine, pro for proline, proline, and HIS for histidine. So these 5 amino acid join Krebs cycle as alpha ketoglutarate, and alpha ketoglutarate ultimately will convert into succinate, and succinate will convert into fumarate, and fumarate into oxaloacetate, and oxaloacetate will produce glucose. So remember these 5 amino acids: arginine, arginine, R, glue, glutamate, and glumine, glutamine, proline and histidine. Arginine, glutamate, glutamine, proline and histidine. These 5 amino acid will join Krebs cycle as alpha ketoglutarate and produce glucose. Now T, Vim's success. Vim's success. So T for threonine, here T for threonine, and V for valine, valine, isoleucine, leucine, and methionine. Methionine. They will join as succinate. Success means succinate. Succinate. See, this threonine comes here also and here also. So threonine can join Kreb cycle as pyruvate and as succinate. So T Wim success. Now toffee 4 means fumarate. Fumarate. So toffee, tyrosine and phenylalanine. So actually phenylalanine and tyrosine, if you see structure wise, they are very similar, same type of amino acid, only hydroxyl group is different. So phenylalanine, they will produce fumarate. They will join Krebs cycle as fumarate, and fumarate will be converted into oxaloacetate, and oxaloacetate will produce glucose. Now AA means aspartate and asparagine. Actually aspartate is keto acid of oxaloacetate. Aspartate, that is keto acid of oxaloacetate. So aspartate and asparagine will join as oxaloacetate. It's now we can, let's go to the previous slide. So gastric will join as pyruvate. Glycine, gastric pylorus. So gastric, alanine, serine, tryptophan, threonine and cysteine, they will join as pylorus, gastric pylorus. Just remember this mnemonics. If you remember this mnemonics, you will remember everything. So this is very important. Gastric pylorus. Now R, arginine, glutamate, glumine provided for his histidine conglomerate. Now third mnemonics is T threonine Wim success, VIMS, valine isoleucine methionine. And fourth mnemonics is toffee, tyrosine, phenylalanine, for me is fumarate. Fifth mnemonics is AA, ox. A is also vowel, you can see ox. You can remember that well. So aspartate and asparagine will produce oxaloacetate. Now glucose will be produced. So this is all about glucogenic amino acid. This is very important topic. So this is all about protein catabolism. So protein catabolism is linked to urea cycle and production of glucose. And that how does this amino acid produce glucose, I have shown in a general perspective. So we will also talk in very detail about this individual amino acid in different lectures. I'll publish those lectures. So remember this very important topic, very important mnemonics. It will work very good for you. And this is the most important about amino acid catabolism. Thank you for watching the video, and see you in next